Hi everyone, welcome to my channel Pure Biology, and today I come with another video that is related to obviously embryogenesis, and here I will try to explain or discuss about the role of the auxin and the cytokinin hormone in the embryogenesis process or plant development process. So this is the third part of the embryogenesis where I will try to explain this role of these hormones. And before this, already two videos related to embryogenesis that I have already uploaded in my channel, so you just check out that too. So here the role of the auxin and the cytokinin hormones is what? The first step during the embryogenic development is the establishment of the apical basal axis. So there are two axis pattern form. One is the apical basal axis and another is the radial pattern form. So here the first, that is the apical basal axis formation, and in which the asymmetric distribution of the auxin mediated by the PIN proteins plays a major role. Okay, major role. So what is saying that the auxin has important role. Why? Because its PIN protein, that is very important, and the distribution, asymmetric distribution, not the same symmetric distribution. The asymmetric distribution is of the auxin, that is important for the development of the apical basal axis. And this asymmetric distribution of the auxin, that is done by the PIN proteins. And the plant polarity is established by the polar auxin transport. So this is the same thing is repeated, that the polarity means the first the shoot apical meristem, and on the other side the root apical meristem. So the two poles that remain in the opposite side, and here the asymmetric distribution of the auxin that play important role for the development of the two poles, that is the apical basal axis pole. Clear? So here one example has taken, that is the Arabidopsis. It has, and it is seen that generally this plant has the two genes. Which are the two genes? One is the GNOM and another is the AMONOPTEROS. That means NOM and monoteros. Two genes are very important that are involved in auxin signaling process. And these NOM genes that encode guanine nucleotide exchange factor, that is short form is GEF, that enables directional transport of auxin. So this signaling process of the auxin signaling process done by the two genes, and the NOM gene that encodes guanine nucleotide exchange factor, that enables the transport of auxin, or directional, which direction is needed for the auxin transport, that is determined by this NOM gene or its product or its factor. And here by auxin, this establishing a polar distribution of PIN auxin efflux carrier protein. So what is saying that this PIN protein, this is very important, and efflux means out of this PIN protein or PIN carrier protein, that plays important role for the polar distribution of the auxin. And it helps in establishing the apical basal polarity. And due to this distribution of the auxin, that polarity of the shoot and root, that will occur. And embryos with in some cases, if the mutation is seen in the NOM gene, in that situation, roots and the cotyledons and fail to develop the axial polarity. Okay, so if NOM gene is mutant, in such cases the roots and cotyledons lack lack of root and cotyledons, it is seen. And another thing is also seen, that fail to develop the axial polarity. That means axial polarity not be developed, and the roots and cotyledons also be absent. Next gene is what? This is a monoterous gene or MP gene that encodes another transcription factor. This is called the ARF or auxin response transcription factor, that mediating that embryo's axis formation. That means two factors here important. One is the GEF factor, another is ARF factor. So these two factors generally important for what? For the axis formation, and another is the vascular development. So in case of NOM gene, the lack of which is happened, roots and cotyledons lack or absent, and the axial polarity will not be developed. And in case of MP gene, monoterous, MP gene's mutation is seen. If the mutation of MP genes is occurred, in that situation also axis formation is not happening, and vascular development also not form or not be seen. Okay. And the seedlings that both the hypocotyle and the root. So another important thing is also said that the mutation of this gene result in the seedlings that both hypocotyle and root. Okay, one thing I have to check first. Okay, I just forgot to write here lack, that's why I have to check this line. So here the mutation, that means the lack of the hypocotyle and the root. Mutations of which gene? The MP gene. Now, product of the MP gene, what will they can do? They generally binds to the auxin response elements in the promoter of certain genes that are transcribed in presence of auxin. So, the gene that transcribed to form, that transcribed when the auxin is present in the medium. In that situation, this gene transcribed. So MP genes, generally product of MP genes, what they do? They generally bind to the factor, that means auxin response factor of the promoter, where in the transcription process. So they generally bind to the auxin response factor of the promoter of certain genes which are responsible for the transcription, or they will transcribe in presence of auxin. Okay, so that means MP genes played important role because it controls the transcription process of such genes which generally transcribe in presence of auxin. Clear? Apical basal patterning of the embryo relies on the differences in the distribution of the auxin across the embryo and through the directed movement of the auxin, and at the 2 cell, this is important here, at the 2 cell stage, the preferential accumulation of the PIN proteins in the apical wall of the basal cell that can be linked with the higher auxin level in the apical cell. So what is saying that when the zygote that divides to form the 2 cell embryo, in that situation the accumulation of PIN proteins, that is very important for the auxin's distribution. So accumulation of PIN proteins, where it is seen? In the apical wall of the basal cell. Okay, in the apical wall of the basal cell, and that is linked with the higher auxin levels in the apical cell. That means where the accumulation is seen, the PIN protein accumulates where? In the apical wall of the basal cell, and that's why the auxin's level rise in these cells. And the distribution is PIN is reversed with higher levels along the basal faces of the apical cells, which in turn leads to higher oxygen level in the basal regions. So first condition, this is, I repeat again, the when the cell stage embryo is formed, the accumulation of pin protein were the apical wall of the basal cell, and that increase the higher oxygen level were in the apical cell. So that times the apical cell will develop. Okay, but in the later or after that, the distribution of the pin that reversed. Okay, when the apical cell development already started, then the process become reversed. And what happened to that? In that situation, pin protein accumulates where the basal faces of apical cells. Okay, in the first condition, apical wall of the basal cell, and here the basal faces of the apical cell, and that's why here basal faces of the apical cells, that in that places or that region, the higher oxygen concentration level is seen. Okay, so in that way, the shoot apical meristem and the root apical meristem both are formed. But when the heart-shaped embryo is formed, in that situation the oxygen level distribution is more complex, and there must be a balance is maintained, the downward internal flow of the oxygen with the upward flow of the oxygen. So here this is too complex when the heart-shaped embryo is formed in the dicotyledons. And now another important hormone is the cytokinin. Its major role is seen in the regulation of the meristem function. And here the important gene is the STM gene. This STM gene is generally NOX gene is very important, and it is related to the NOX gene obviously. The NOX gene has two subclasses, NOX1 and KNOX. NOX1 and NOX2, and STM gene that belongs to NOX1. Okay, belongs to this. And this STM gene that encodes STM protein, and which induces the cytokinin biosynthesis in SAM. SAM means shoot apical meristem. So here the STM protein, very important, which encodes this STM gene to control the cytokinin biosynthesis for the development of the shoot apical meristem. Clear? And here another important thing is that the product of NOX gene also control the biosynthesis of the GA. GA means gibberellin hormone, and it represses the biosynthesis of gibberellin hormone when it controls the biosynthesis of the cytokinin hormone for the development of the shoot apical meristem. And here the GA level that become decreased, and in that situation maintained meristem functions, that is controlled by the GA hormone or gibberellin hormone. And it also stimulates the genes that is involved in the GA catabolism. Catabolism means GA hormones biosynthesis when increased, that is called the catabolism. And to reinforce the low GA level established by the NOX protein in the assay. So main important thing is that the cytokinin plays two major roles in case of gibberellin hormone. That means it controls the catabolism process of the gibberellin hormone and also the anabolism process of the gibberellin hormone. That means the represses the biosynthesis of the GA and the also catabolize the biosynthesis of the GA. And here when GA is needed for the development of the meristematic tissues, then GA level that will rise. And when GA level is not needed at the time when the shoot apical meristem development is occurring by the hormone of the cytokinin, in that situation the GA level that become very low. Okay, so this is the process of the shoot apical meristem and root apical meristem, and how this controlled by the two main hormones, auxins and cytokinin. So if you like the topics, so please do share and subscribe this channel and to support this channel. Okay, thank you.